Hello students, welcome you all to a new video of Sunanda's tutorial. In this video, we will discuss about protein folding. So at first, we have to discuss what are different structures of protein. So protein is manufactured by two processes in two steps, transcription and translation. After translation, linear chain, sorry, linear chain like this, linear polypeptide chain of amino acids is formed. So different types of amino acids attach with each other by forming peptide bond to form primary structure of protein after translation. After primary structure, they fold to form secondary, then to tertiary, then to quaternary. To play a specific role, it have to fold. So today we will discuss about folding, how this protein folding takes place. So protein folding is a physical process, often spontaneous process, because it occurs automatically where a linear polypeptide chain, which is the result of translation, twist, folds into specific stable 3D shape, is known as native state of that particular protein, which is crucial for its biological function. So to play a particular function, the protein has to fold into a specific shape, forming a specific shape, 3D shape, called as its native state. Driven by amino acid interactions, primarily hydrophobic effect, this process converts a random coil into functional protein. So what is the main force which plays for protein folding? It is hydrophobic effect, and it converts the random coil of protein into a functional protein, into a specific shape with correct folding essential, as misfolding leads to disease. So it is the basic idea about protein folding. So what are different structures? Where are the hierarchical sequence of structures? At first, primary structure of protein, then secondary structure like alpha helix and beta acid. So different types of structure we will discuss in my future videos. We will discuss about different structures of proteins, but today we will discuss only about protein folding. So secondary structure, primary linear sequence of amino acids fold to form alpha helix or beta acid or beta plates, which are the secondary structure of proteins. Then tertiary, which is a complete 3D conformation of a single polypeptide chain driven by interaction between side chains, R groups. Quaternary structure, the arrangement of multiple folded polypeptide subunit into single functional complex. For example, hemoglobin. Hemoglobin is a single functional complex, and which is the result of the folding of different polypeptide subunits, which are the aggregation of different polypeptide subunits, results in the formation of quaternary structure. Example, hemoglobin. Okay, next. So it is the structural representation of how primary protein fold to form secondary structure of protein, then tertiary, then finally quaternary. So key driving force, at first, hydrophobic effect. Hydrophobic effect, it is the primary driver where non-polar amino acids cluster in proteins interior, cluster in the proteins interior to avoid surrounding water. Hydrophobic means water hating. Okay, then what is hydrophobic effect? At first we have to remember what is non-polar amino acids. What is non-polar amino acids? So those protein who have uh, high aromatic or any hydrocarbon in their side chain. So aromatic compound or hydrocarbon cannot form hydrogen bond with water, so they are insoluble in water. That's why they have to cluster or aggregate in the interior surface of proteins. Okay, to avoid the surrounding water, because they cannot make hydrogen bond with water molecule. Okay. Then hydrogen bonds are another key driving force formed between polar side chains or backbone atoms to stabilize secondary and tertiary patterns. Then ionic bond or salt bridge, attraction between positively and negatively charged side chains of amino acids. Disulfide bridge, strong covalent bonds between cysteine residue that lock parts of the proteins together. So these are some driving forces for forming different types of structures, 2D, 3D protein structures. Which molecule controls the whole process of protein folding? It is the most important part. While some protein fold independently, many require help to avoid kinetic traps or incorrect aggregation. So molecular chaperone is the most special protein that binds to nascent or misfolded chain to prevent aggregation and facilitate correct folding. Chaperonins, which are large barrel-shaped complex that provide an isolated cage for single protein to fold without interference from other crowded or other environments, other proteins. So it acts as a protector. It protects the protein from other proteins or other disturbances of the environment. Then folding catalyst, enzymes like protein disulfide isomerase, peptidyl propylyl isomerase that speed up slow steps in the folding pathway. So these are some proteins, some enzymes which assist or regulate the process of protein folding. Okay, next, medical and scientific significance. When protein fails to fold correctly or misfolding takes place, they can form toxic aggregates called amyloid fibrils. These are linked to several serious diseases like neurodegenerative disease like Alzheimer's disease, Parkinson's disease, Huntington's disease, Bryan's disease, other disorders like cystic fibrosis, various allergies. These are caused by misfolding of proteins. Next, why this protein folding is important? At first, to play a specific role, then disease prevention, biological regulation, then in medical research and biotechnology, it is very crucial to develop a targeted drug. Okay, then in AI and modeling to determine how proteins fold allows for prediction of protein structure, then immune system interactions. Okay, because incorrectly folded proteins can lead to immune system error or disorder such as causing allergies and other diseases. So this is why protein folding is crucial for our body to function correctly. So it is all about protein folding. I think you will get benefit from this video. If you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.